Thank you.
Thank you for the introduction. So today I'm going to talk about the rational combination therapy of mechanical, HIFU, and immune checkpoint blockade. So HIFU is known as a non-invasive treatment modality for localized tumors, but it also induces anti-tumor immune responses by causing immunogenic cell death of tumors. However, local recurrence and distant metastases still remain major challenges of local therapies. Today, I'll illustrate differences between thermal and mechanical HIFU and induction of immune responses and anti-tumor efficacy. So we use human HER2-expressing murine breast cancer cells, MM3MG HER2, in immune-competent mice, as shown in this schema. Tumor cells were subcutaneously injected to mice, and tumors were treated by T or M HIFU using VIFU 2000 system. The optimized protocols of T and M HIFU are shown in this table. So as you can see by images of the cut surface of treated tumors, T HIFU caused coagulative necrosis in the tumor, while M HIFU induced merely local destruction of the tumor tissue through acoustic cavitation. In these experiments, M HIFU significantly suppressed tumor growth compared to T HIFU, and 40% of the animals showed complete remission, resulting in improved survival of mice. And we also measured the circulating HER2-specific immune response induced by HIFU. Interferon gamma ELISpot data demonstrated that M HIFU induced significantly stronger HER2-specific cellular immune response compared to T HIFU, which was consistent with the tumor growth data. Next, we analyzed tumor-infiltrating immune cells. Flow cytometric analysis of the tumor infiltrates revealed that M HIFU enhanced tumor infiltration of CD4 and CD8 positive T cells and NK cells. Finally, CD8 positive T cells in M HIFU treated tumor showed high activation and cytotoxic function. And PD-L1 expression by tumor cells and immunes are known to represent key immune checkpoints. Therefore, we measured PD-L1 expression on both cell types after treatment. And we found that M HIFU induced increased PD-L1 expression, not only on tumor cells, but also on the immune cells. And we utilized single-cell RNA-seq analysis to allow further immunological characterization in tumor-infiltrating immune cells, as shown in this schema. Single-cell RNA-seq analysis of immune cells following HIFU's treatment demonstrated elevated expression of co-stimulatory and cytotoxic markers of CD8 positive lymphocytes in M HIFU treated group. And in contrast, the CD8 positive lymphocyte population was decreased in the T HIFU treated group. And based on these results, we hypothesized that combining immune checkpoint blockade to M HIFU treatment would enhance the anti-tumor efficacy. As shown in the experimental schema, we treated mice with anti-PD-L1 antibody injection after initial M HIFU treatment. So as shown on your left, the combination treatment prolonged survival compared to monotherapies and achieved 72% curative rate. Additionally, we confirmed the strongest HER2-specific cellular immune response in combination strategy using ELISpot assay. So in conclusion, M HIFU treatment demonstrated significantly suppressed tumor growth by inducing anti-tumor cellular immune response. Single-cell RNA sequencing revealed the expression level of immune checkpoint-related genes and could be a rational proof for the combination strategy of local therapy and immune checkpoint blockade. Combining HIFU and anti-PD-L1 antibody demonstrated enhanced anti-tumor cellular response and prolonged survival of tumor-bearing mice. So I would like to briefly acknowledge our collaborators, Dr. Song and Dr. Robertson, and the staff member in our lab. Thank you for your attention.
All right, this paper is open for questions. We may have a question coming. Or maybe not. So I have a question for you. Could you tell us more about your protocols? I would love your acoustic pressure, but also, do you know the temperature that you were achieving in the thermal, and what fraction of the tumor were you treating?
Sorry?
What fraction, like what volume fraction of the tumor were you ablating, for example?
Okay, so sorry, I skipped pretty rapidly. So this is a protocol, and sorry, I didn't write the pressure, but I thought that was 6 megapascal or so. And the actual temperature we measured was 64 degrees in the T-HIFU treatment, whereas 71 at the mechanical HIFU. And usually we treat the tumor around the diameter of 6 or 7 millimeters. And also we treated 6 parts as shown in the figure.
So in each case, you distributed, it looks like 9 points, 9 foci throughout the tumor volume?
Right.
Is that correct? Great. Other questions? All right. Thank you so much.